Okay, let's continue for integration of metabolism. So just recap again, for us to understand how metabolic processes integrate with each other. So there are three states in our body that we can look into as an example of how these processes relates. Okay, so we have fat state, or where we are full after we have eaten something, for example. Okay, and then second we have fasting state. And the next one, which is not here, is actually the starvation or prolonged fasting. Okay. Okay, so this is generally what are the processes or the reactions that takes place during fat state and also fasting state. So for starvation state or prolonged fasting is quite similar to fasting state. Okay, but we will look into what are the differences later. So for fat state is during after we have taken our dietary intake, for example. So the processes that takes place during fat state would be glycolysis, the breakdown of glucose, and then we have glycogenesis, storage of excess glucose. We also have lipogenesis, okay, and then protein synthesis. So the hormones that are actively involved or triggered or activated in fat state would be insulin. Okay, so in fasting state, the processes or the reactions that takes place are gluconeogenesis, so the production of the synthesis of glucose from non-carbohydrate sources. We also have glycogenolysis, meaning the breakdown of glycogen into glucose. We have lipolysis, ketogenesis and ketolysis, and also protein breakdown. So the hormones involved here, it would be glucagon, epinephrine or adrenaline, growth hormone, and also cortisol. Okay, so for us to remember, so remember that for fat state, we have excess glucose in our bloodstream. So we are trying to utilize it and also store it. Whereas for fasting state, there's a reduction of blood glucose level. So we are trying to increase the blood glucose level, right? Alright, so for this is for fat state. So we will look into fat state one by one. So during fat state, so remember, fat state is when after you have taken something in your diet lah, in dietary intake. Okay, so the insulin can reduce the blood glucose by increasing the uptake of glucose in adipose tissue and muscle. Okay, so after eating, blood glucose level increase. So now the insulin will try to reduce the blood glucose level. So what it will do, it will increase the uptake of glucose into adipose tissue or muscle tissue. Okay, so how will insulin do this is by increasing the number of glucose transporter. Okay, so for example, the green color would be hepatocytes. So insulin will trigger the activation or increase the expression level of glucose transporter. So what will this glucose transporter do? It will cause the increase of uptake of glucose from the bloodstream into our hepatocytes. Okay. So once the glucose has entered, for example, the hepatocytes or the skeletal muscle tissues, so glycolysis will take place. So from glucose, now glucose will be converted into glucose 6-phosphate and so on lah, the glycolysis reaction. Okay, so in glycolysis, it will convert one molecule of glucose into 2 molecules of pyruvate. So this is also important for us to remember. And all this reaction takes place in the cytosol. Okay, so I won't go through all these details. You can go into it later. Okay, so these are the important enzymes in glycolysis. Alright, so glycolysis have also regulation in terms of its enzyme. For example, we have allosteric regulation, meaning the process of the reaction controlled by other molecules or even molecules lah basically. For example, fructose 2,6 bisphosphate, AMP, ATP, meaning that, for example, if there's an increase or upregulated level of ATP, this will control or decide whether or not this process will be continue or not. Okay, so that's allosteric regulation. We also have covalent modification or for example, phosphorylation of enzyme. So for example, if there's phosphorylation of phosphofructokinase over here, it will alter the morphology or the structure of this enzyme, which could then inhibit, for example, the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Okay. Okay. So now again together, okay. After we have eaten, increase in blood glucose level. So what our body will do, the insulin will now trigger the upregulation of glucose transporter. So this is the transporter. So this transporter will cause an uptake of glucose into our cells, for example, the liver cells. So inside the liver cells, glycolysis will take place. Okay, so glycolysis involve a lot of enzyme because they have a lot of intermediates. So these enzymes are regulated by these two mechanisms, allosteric regulation and also covalent modification. Allosteric means they are regulated by other molecules like ATP, while covalent modification involve, for example, the phosphorylation of the enzyme itself. Okay, so that's one regulation. Okay, I won't go into detail. So we also have anaerobic glycolysis, which also takes place within the hepatocytes. Okay, so in anaerobic, instead of pyruvate is formed, our body will form lactate. Okay, for example, intense exercise. Okay, so this lactate will be released into the bloodstream bag and then taken up by the liver to convert it to pyruvate and also glucose for us to utilize the energy. Okay, so I won't go into detail for this one. Okay, so for example, just now, so for glycolysis, remember, it won't end up until pyruvate. So this pyruvate will will be converted into acetyl-CoA, and this acetyl-CoA will enter the Krebs cycle. So this is where more ATP would be produced. ATP means more energy will be produced for us to utilize, right? Okay, so this is glycolysis and then followed by Krebs cycle or TCA cycle. Okay, so again, after glycolysis that happens in the cytosol, so Krebs cycle happens in the mitochondria. Okay, this one you can go into later. All right. Okay, so remember again, in fat state, we have covered glycolysis. So during fat state, high glucose in the blood, so glycolysis will take place because we need to break down the glucose that has been in our diet. Okay, so next process would be glycogenesis. So remember, as the name suggests, okay, glycogenesis meaning the synthesis of glycogen. So what is glycogen? Glycogen is the storage form of glucose. Okay, so now that we have taken a lot of glucose in our diet, it needs to be stored, right? So that's when glycogenesis takes place. So where glycogenesis takes place is within the cytosol. Okay, and the important enzyme would be glycogen synthase. Okay, or we call it as rate limiting step. And this is also activated by insulin. Okay, so insulin will send signal to our body. Now you have extra glucose in your body. So we need to do glycogenesis to store the excess glucose. Okay, so that is for glycogenesis. This is pentose phosphate pathway, which we would be covering in this semester. But just for you to know, glucose can also enter this pentose phosphate pathway. Okay. Again, similarly for the generation of energy, right? Okay, so during fat state, we won't be only eating carbohydrates meal, right? So we also eat things that are fatty or even have protein sources. Okay, so in fat state, we have fatty acid synthesis and also lipogenesis. Okay, the increase in glucose and insulin level will stimulate the synthesis of fatty acid and also triacylglycerol, or what we call as lipogenesis. Okay, right. So this fatty acid synthesis takes place within the cytosol. So if you see over here, we have the green colored one is the hepatocytes. The orange color cells here is the adipocyte or the fatty tissue. Okay, so we have glucose metabolism happening as well over here, the glycolysis and also Krebs cycle. But we also have fatty acid synthesis over here. Okay. Okay. So the important enzyme for fatty acid synthesis and also lipogenesis would be acetyl-CoA carboxylase and also fatty acid synthase, right? So we will use the free fatty acid, combine it, for example, with glycerol. So this is just an example, and it will be transported in the form of lipoprotein, for example, to the adipocytes to be stored. Okay. All right. So this is for lipogenesis. We also have cholesterol synthesis. So again, bringing you back to the initial idea of fat state. When we eat, that's when our body will use that opportunity to take out whatever that we have in our meal, for example, to use it to reconstruct back things in our body. Okay, so if the cholesterol level is depleted, then this is the time our body will use that whatever that we have taken from our meal to construct or to build or to catabolize the formation of cholesterol, for example. Okay, so again, similarly for cholesterol synthesis, it also takes place in the cytosol, and the important enzyme would be HMG-CoA reductase. Okay, and this is activated by insulin. Okay, because insulin is the main actor here in terms of hormone, because remember fat state, and inhibited by drugs, for example, statin, okay, anti-cholesterol drug. All right. Okay, so next would be the amino acid metabolism during fat state. So different from fatty acid and also glucose. So the amino acid, they don't have, we don't have the capability to store amino acid in our body. So the amount that we have eaten must be utilized to make, for example, structural proteins, or to make enzymes, or to make even to make glucose, gluconeogenesis. So the remaining or the excess amino acid will be excreted out or removed in the form of urea. Okay. So that is for amino acid. Okay, I won't go through in detail for this one. This is the process. Okay, so that's about it for fat state. So remember, for fat state is when the glucose level is high in our bloodstream because we have consumed something. All right. So when the blood glucose level increase, insulin will be activated and released. So this insulin will trigger the metabolic reactions of other things. For example, the glycolysis, which will move into Krebs cycle. We also have fatty acid synthesis and also lipogenesis. And then the amino acids will also be used to make proteins, enzymes, for example, in our body, and the remaining would be removed in the form of urine. Okay. All right. So that is for fat state. Okay. So these are the processes that are happening during fat state, or when we have, after we have eaten something. Okay. Do you have any question for fat state? Okay, let's take a 2 minutes break for you to digest the information. Okay, can we come back at 2:50? Okay. All right. So can we continue?
Yes.
Okay, so we have covered just now fat state. So now let's look into fasting state. So fasting state is generally the opposite of fat state. If fat state is all about high glucose level in blood, so in fasting state is where we have low or reduced blood glucose level. Okay, so these are the important hormones in fasting state, which are glucagon, adrenaline or these epinephrines, growth hormone, and also cortisol. Okay, but you can only focus on this glucagon and also adrenaline. All right. Okay, so in fat state, it's about taking up the glucose that is inside our blood and then absorb it or uptake it into our cells. For example, the liver cells, all right, and for storage. But for fasting state is where we want to increase the blood glucose level back, right? So the first metabolism here would be glycogenolysis. So glycogenolysis, meaning the lysis of glycogen or the breakdown of glycogen. So what is glycogen? Glycogen is the storage form of glucose. So we want to break down the storage form of glucose, which is the glycogen, back into glucose. So this reaction takes place in the cytosol, and the important enzyme would be glycogen phosphorylase. Okay, so if in fat state, insulin plays an important role. So in fasting state, the important hormone here would be glucagon and also adrenaline. Okay, as we have covered before this in our tutorial. So adrenaline is important in fasting state or even in condition where blood glucose level is low. For example, during intense exercise. So adrenaline will trigger for our body to produce more and more blood glucose level. Same goes to glucagon. So when our body sense that the blood glucose level is low, that is when glucagon is also activated for further metabolic reactions to take place. Okay. Okay, so glycogenesis occurs both in liver and also muscle because as I've mentioned, okay, for example, when there's involvement of adrenaline, this shows that our body is in active state or intense exercise. So this process needs to occur quickly or fast. So we need the instant source of glucose. So that's why it can occur both in liver and also muscle, right? So that is glycogenolysis to break down the storage of glucose. So what if it's not sufficient? That's when gluconeogenesis will take place. So gluconeogenesis is the production or the synthesis of glucose from non-carbohydrate sources. So meaning we are producing glucose from, for example, fatty acid, amino acid, right? Okay, because glycogen or the storage form of glucose is easily used up. So gluconeogenesis is important to maintain the blood glucose level. Okay, so again, it's all about maintaining or increasing back the blood glucose level. Okay, so for example, glycerol, lactate, and also amino acids will be taken up into, for example, the hepatocytes or the liver cells for them to be enter the glycolysis metabolism. Okay, so for example, amino acids, they can be intermediates of Krebs cycle. So this will increase, for example, the energy production or ATP production. All right, I will go through this in detail as well. Okay, so that is gluconeogenesis. The important enzymes would be pyruvate carboxylase, PP carboxykinase, fructose 1,6 biphosphatase here over here, glucose 6 phosphatase. All right. Okay, so that is gluconeogenesis. So if in fat state, the reaction for fatty acid would be lipogenesis, right? But for in fasting state, it's all about lipolysis, breaking down fatty acid and also beta oxidation of fatty acid. Okay. So this process again, it takes place in the mitochondria, and the important enzyme would be carnitine acyltransferase 1. Okay. So sorry. Okay. So in fasting state will be lipolysis and beta oxidation of fatty acid. Okay. We also have this condition where our body will start to use ketone bodies. right? So when there's insufficient of glucose, and even gluconeogenesis wouldn't be able to increase back the glucose level in our body. So that's when our body will start to utilize ketone bodies as its fuel of energy. Okay, so this one is within the lipid metabolism, you can go through later. Okay, so in fasting, ketogenesis also takes place. Okay, as for amino acids, right, the carbon skeletons of amino acids are an important source of substrate for gluconeogenesis. So they are derived from the protein breakdown as a result of hormone, for example, corticosteroid action. Okay. Oh, so in fasting state for amino acids, our source of amino acids could be taken from the protein that has been broken down. Okay, so that's why this is also related to ketone bodies as well. Okay, and also yes, protein breakdown. Okay. Let me share you the lecture note. Yep. Okay, so just now we actually have gone into a bit of prolonged starvation. All right. So after fasting state, if that condition prolongs, that is when we call it is as prolonged starvation. So to provide glucose for the brain, the liver will start to degrade certain protein. So those most expendable in an organism not ingesting food. Okay, they are non-essential amino acids are transaminated or deaminated. So the extra amino groups are converted to urea, which is exported by the bloodstream to the kidney and excreted. So point number one would be, this is how you read this diagram. Point number one is here, number one here. So protein degradation, this will yield glucogenic amino acids. So point number two, so if there's an extra of this is not utilized, then it will form a lot of urea, and this urea will be exported to the kidney and excreted in the urine. Okay, so the carbon skeletons of glucogenic amino acids are converted into pyruvates. So these amino acids will then be converted intermediates of the Krebs cycle, for example, right? So these intermediates, as well as the glycerol derived from the triacylglycerols in the adipose tissue, provide the starting material for gluconeogenesis. So along with amino acids converting into the intermediates of Krebs cycle, the same thing happens with glycerol, for example. So they are also being converted into intermediates of Krebs cycle. So this is for gluconeogenesis. Okay, so eventually the use of citric acid cycle intermediates. So if these intermediates are all used up for gluconeogenesis, this will, for example, over here will deplete or reduce the amount of oxaloacetate. So if oxaloacetate is reduced, then there will be inhibition of acetyl-CoA into the citric acid cycle. Okay. So acetyl-CoA produced by fatty acid oxidation now will accumulate because they cannot enter the Krebs cycle. Okay, so if this acetyl-CoA accumulate, they will now favor the formation of acetoacetyl-CoA and ketone bodies in the liver. So point number 7. Okay, so acetyl-CoA accumulation favors ketone body synthesis. So after a few days of fasting, the level of ketone bodies will increase. Okay, remember that when our body is not capable of producing back or increasing back the blood glucose level, it will start to shift to production of ketone bodies. So the organs, the vital organs will start to use ketone bodies as their fuel of energy. Okay, so these ketone bodies are then exported to, for example, heart and brain, which we go through just now, which use them instead of glucose. Okay, So this is how the changes of each what we call as source of energy happens in our body during prolonged starvation. So despite the hormonal mechanisms for maintaining the level of glucose, it will begin to diminish after 2 days of fasting. Okay, so after 2 days of fasting, now we have entered the prolonged starvation. This is not the normal fasting that we have during Ramadan. Okay, that's when we break our fast, then we eat a lot of things, so that is not prolonged starvation. So prolonged starvation is mean continuous uh, continuous apa lack of food lah. So we haven't eaten anything for 2 days. Okay, so in that condition, even our hormones are unable to restore back the glucose level. Okay, so the level of ketone bodies, almost immeasurable before the fast, will rise dramatically after 2 to 4 days of fasting or prolonged starvation. So insulin will no no glucagon will are not able to bring back the glucose level back to normal. So now our body will shift to ketone bodies utilization. Okay, so these water soluble ketones, acetoacetate and also beta hydroxybutyrate. So ketones, acetoacetate and also beta hydroxybutyrate are all ketone bodies. Okay, supplement glucose as an energy source during long fast or during prolonged starvation. Okay, so that's why after few days of fasting, glucose level will continue to drop. And if we haven't eaten anything, same goes to fatty acids. The level will be constant, but ketone bodies will increase. Okay, and vital organs will start to use ketone bodies instead of glucose. Alright. Okay, so that is all about integration of metabolism. Okay, so if you go back to the extended notes earlier, okay, so remember to understand how these metabolic reactions takes place in our body and regulates with each other. We have observed three conditions in our body: the fat state, fasting state, and also prolonged starvation. Okay, so in fat state, meaning when we are full after we have eaten our food, so that's when the blood glucose level is high. Alright, so the goal here is to reduce the blood glucose level. So how do we do that? We will break down the glucose, we will store the glucose, and we will convert them into, for example, um, uh, storage lah, for storage. Okay, and then for ATP as well. And in terms of our meal, we don't only consume carbohydrates sources, we also have proteins and also fat sources. So what will happen to them is we will start to utilize them for energy, for example, or for storage, alright, or for us to develop or reconstruct back, for example, our enzymes or our tissues, to repair our tissues, for example. Okay, so the important hormones here for fat state would be insulin. Okay, so during fasting state, so the key here in fasting state is when our blood glucose level is very low. So the goal of our body is to increase back the blood glucose to normal level. So what we will do, we will produce or synthesis new glucose molecule from, for example, gluconeogenesis from amino acids. Or we can even break down glycogen, the storage form of glucose. So this is through glycogenolysis. Okay, and then we will also break down lipids through lipolysis so that we can utilize them for energy. Right, and this lipolysis can form acetyl-CoA, for example. And then we also have in prolonged starvation, we have ketogenesis, okay, formation of ketone bodies. And we also have protein breakdown in our liver. So in fasting state or the prolonged starvation, the important hormones would be mainly glucagon. So, okay, the opposite of insulin, but we also have adrenaline, growth hormone, and also cortisol. Correct. So that's about integration of metabolism. Do you have any question? So this chapter is generally a recap of all the metabolic reactions that we have go through, but we are trying to see how this integrates according to this condition. Okay, do you have a question? Tak ada? All good?
Not yet.
Alright, if you don't have any question, then we will end our class for today. I will upload the recording later. Alright, thank you everyone.
Thank you, Miss.
Thank you, madam. Thank you, madam.